All right. Good morning, everyone, and thank you for coming to my talk on the work that I did during my MSc. So monocytes and macrophages are important cells of the innate immune system, which is the body's first line of defense against infection and tissue damage. So monocytes patrol in the blood, and they migrate into infected or damaged tissue, where they differentiate into macrophages. These macrophages then act as specialized phagocytes in those infected tissues. Studying monocyte-to-macrophage differentiation in vitro usually involves pro-monocytic cell lines such as THP1s. These cells can be treated with PMA to induce differentiation into macrophages, and these THP1s, as well as the PMA-stimulated macrophages, have been shown to be similar to primary monocytes and macrophages. The process of monocyte-to-macrophage differentiation is associated with transcriptomic changes. However, changes in alternative splicing have not been well characterized, although some studies have shown that alternative splicing does regulate this process, and RNA binding proteins prime cells to respond to stimuli. So what is alternative splicing? It is a mechanism in which a single pre-mRNA transcript results in multiple mature mRNA isoforms. Alternative splicing occurs in more than 90% of multi-exonic genes and results in a significant increase in protein and transcript diversity. Some of the most common alternative splicing events are shown on the screen. So how do we analyze alternative splicing? This analysis is broadly categorized into isoform-based and count-based methods, where isoform-based methods involve the full reconstruction of isoforms from RNA sequencing reads and the subsequent quantification of the expression of these isoforms. Count-based methods construct genes into disjointed counting bins that are inferred from isoform-specific reads, where we can then quantify the expression of transcript features, which can include exons or splice junctions. Count-based methods have been shown to be more appropriate when analyzing short-read RNA sequencing data, as these methods provide a more straightforward analysis, and they avoid the challenges that are associated with constructing full isoforms using short reads. Some of the key count-based methods that are used to analyze alternative splicing include differential exon usage, differential transcript usage, and differential alternative splicing event analysis. DEU measures the differences in the expression of exons within genes between conditions and provides information on which exons are included or excluded in genes between our condition groups. DTU measures differences in the proportion of transcript abundances of all transcripts within a gene between our conditions, whereas DAS analyses detects alternative splicing events and quantifies their percent spliced in or PSI values, which is a measure of the inclusion of an exon within a transcript. These PSI values, DAS analysis then measures the differences or the changes in these PSI values before each detected event between our condition groups. I therefore use these count-based methods to investigate alternative splicing changes between monocytes and macrophages using a lab-established protocol where we use low-dose PMA to induce THP1 monocytes to differentiate into macrophages. So I performed quality control on the RNA sequencing reads, and once I established that these reads were of high quality, I aligned them to the human reference genome using the splice-aware aligner HiSET2. The output files from HiSET2 were then inputted into DEXseq, where it calculated the statistical significance of the differences in the counts of one exon or exonic part compared to all the other exons within genes between our monocytes and macrophages. DEXseq identified 451 differentially used exons affecting 160 genes. Transcript level quantification was performed using RSEM to calculate the estimated transcript abundances in our monocytes and our macrophages. These estimated abundances were then used by DRIMseq to calculate the statistical significance in the differences between these abundances between our monocytes and macrophages. As DRIMseq does not have the appropriate statistical power to infer differential transcript usage, as it performs this analysis at both a gene and a transcript level, stage-wise analysis was performed using stage R, where the p-values for the gene level were screened and then a statistical analysis was performed on the transcript level p-values in order to confirm that these genes do in fact experience differential transcript usage. Stage R identified 671 differentially used transcripts affecting 211 genes. And lastly, the BAM files that were taken from HiSET2 were inputted into RMATS Turbo, where RMATS detected the alternative splicing events that occur between the monocytes and the macrophages and measured these PSI values and calculated the statistical significance of these changes in PSI values for all the alternative splicing events detected between our monocytes and macrophages. 238 differential alternative splicing events were identified, affecting 196 genes. And so then what I attempted to do was to integrate all the results from each of these methods in order to look at the different levels of alternative splicing simultaneously. However, we see that there is limited overlap with only one gene experiencing DEU, DTU, and DAS. This gene is visualized on the right, and it is PBRM1, which encodes for a subunit of ATP-dependent chromatin remodeling complexes. Studies have shown that this subunit interacts with transcription factors that are important for monocyte-to-macrophage differentiation, and that this gene is involved in epigenetic modifications of macrophage markers. We can see here that the differential usage of this particular exon is expressed more in the macrophages, as it is purple, than in the monocytes, and we see that this has a slight effect on the isoform switching that is experienced, as for some of these transcripts, the expression of these is lower in the macrophages than monocytes. Now, to look at a different part of this Venn diagram, we look at the overlap between DEU and DTU, which is absent in DAS, and this gene is the SHC1 gene, which is involved in pathways that are activated during differentiation, more importantly, monocyte-to-macrophage differentiation. Again, we see that these exonic parts are differentially used with higher usage in the macrophages, and that also has a similar effect on the isoform switches. And therefore, we can conclude that there are significant alternative splicing changes as monocytes differentiate into macrophages. We also see that between count-based methods, there's limited overlap due to differences in methodology and the statistical analysis performed by each of these tools, and also because there are different regulatory mechanisms that control alternative splicing at different levels. However, we still see that these individual approaches can still provide insights into the global alternative splicing landscape, and it has revealed that alternative splicing plays a role directly in the differentiation of monocytes into macrophages. I would like to thank everyone for listening to my talk, and I would like to thank my supervisors and my lab mates and their lab, as well as my funding bodies. I was doing short reads. Yes. So these particular methods have been specifically designed to essentially bridge over the splice junction. So they focus more on the features of the transcript instead of the transcript as a whole. So count-based methods essentially count reads that align to splice junction specifically and to exon specifically, rather than trying to look at the transcript as an entire thing. And so once you look at these reads, and you can essentially summarize those counts that align to those different features, and you can then look at the quantification of those or the differences in those features specifically instead of looking at entire transcripts. Hi. So for my particular project, I didn't look at the differential gene expression. There are some members of my lab who are looking into that. And so the next step is essentially to integrate all of these and to look at it at a multi-omics perspective. So essentially what we planned was to kind of look at these things differently or separately so that we don't kind of infer information from another type of analysis, especially because we've seen that the changes in gene expression don't reflect as changes in alternative splicing. So there are differences in the expression of macrophage markers. However, there aren't significant splicing in those to show that there's different regulatory things. And so what we see here that once we look at the different features specifically, we can then assemble these kind of transcripts. Well, not assemble per se, kind of like a pseudo assembly, because it's hard to kind of make a full assembly. And we can then quantify the expression from those abundances in order to see if there's a switch in the transcripts within a gene and between or across conditions. I hope that answers your question.